This is the last section of the carbohydrate chapter. We're going to talk about polysaccharides. Again, this is my cartoon version of a polysaccharide, and you probably understand why this is the cartoon version now. Polysaccharides are long chains of monosaccharides. So here's one monosaccharide attached to another one, attached to another, and on and on and on. So if you attach many of them, you make a polymer of monosaccharides, which is why polysaccharides are called polysaccharides. These pictures here are just three different materials that are predominantly made of polysaccharides. So this is cotton, and cotton is the white fluffy stuff is pretty much polymer of monosaccharides. It's a polysaccharide. This is our U.S. currency. Our paper money is pretty much made of cotton. I actually used to make this stuff when I worked in a paper mill. I made paper money, believe it or not. And potatoes. Potatoes are predominantly made of polysaccharides as well, especially the stuff inside of the peel. So that's sort of a brief introduction to polysaccharides. There are many different types of polysaccharides out there in the real world. Some are made of polymers of glucose. Some are made of polymers of fructose. If you can think of a monosaccharide, there is probably a polysaccharide out there that is made of it. Sometimes the polysaccharides sort of have one long chain, and I was showing you an example of that here. This is just sort of a cartoon version of a polysaccharide that has one long chain. That's not always true. Sometimes polysaccharides can have branches in the chain, so they can have sort of multiple chains chained together, if you want to think about it that way. Very often polysaccharides are used for long-term energy storage. So if you've ingested too much sugar and you have more than enough for your energy needs, you can sort of attach them to each other and then sort of shove them in the corner of a cell and sort of keep them there for some other time in the future when you need the energy, when you might need the energy. So polysaccharides are very often used for long-term energy storage. So again, as I show here, if you eat too much mono or disaccharide, your body can attach those monosaccharides and disaccharides to each other and sort of store the polysaccharide away. Let's see. And again, I don't expect you to memorize these chemical structures, but this is just sort of going to be a short parade of different types of polysaccharides. This particular polysaccharide is called amylose. It is sort of one long chain with no branches. This is just one type of polysaccharide. This is a dominant polysaccharide. A large part of potatoes is made of amylose polysaccharide, and this is basically glucose molecules attached to each other over and over again in the same way. And you can see here that the bonds are pointing down at the connection point, and the connection point is this oxygen. Because the bonds are pointing down, that means that both of the alcohol groups for this monosaccharide and for this monosaccharide were both pointing down when the attachment was made. And it has a fancy name. It's called an alpha-1,4 glycosidic bond. You don't need to know what that means. I can explain it very briefly. Basically, this is carbon number 1 of this molecule, so that's where that 1 comes from. This is carbon number 4 of the other monosaccharide that was being attached, so it's called a 1,4 bond. Glycosidic just means a bond made between monosaccharides or glucose monosaccharides. And alpha just means that both of the alcohol groups were pointing in the down position. They were both pointing in the alpha position when the bond was made, but you don't need to know how that naming comes along. You just need to be able to look at this and say, yep, both alcohol groups were pointing in the down position. And when you do that over and over again and attach them in that way, you make a polysaccharide called amylose. This is also found in crackers and things like that. Amylopectin is a different polysaccharide. Again, pretty much made of glucose, except that it has branches. So you can have long chains of glucoses attached, sort of shown here, and then there can be a branch either to the north or to the south, and then you can start a new chain. And this is amylopectin. This is a polysaccharide that's found in many fruits and other foods like that. And I want to point out one other thing. This is the oxygen that is sort of attaching two glucoses. Here's one, what used to be a glucose monosaccharide attached to what used to be another glucose monosaccharide, and they're attached at this oxygen point. This is just another way, this little U here, starting going down, then to the right, and then going back up. This is just another convention for pointing out to people that both alcohol groups were pointing in the down position when the attachment was made. So again, this is how, if you were to see these chains of glucoses attached in this way with the occasional branch, you probably have the amylopectin polysaccharide. But this is just an example of one with a branch. There's glycogen. This is one that I'd like you to be more familiar with. It's a polysaccharide used by humans and other animals to store monosaccharides. It's a lot like amylopectin here, except it's similar to amylopectin, but it has more frequent branches, so it has more branches. But it's a lot like the molecule that I showed you on the previous slide. You can have thousands, probably tens of thousands of glucose monosaccharides attached to each other. And the reason I want you to know what glycogen is because it's one of the molecules that is used by humans for long-term energy storage. You eat too much sugar, some of that sugar is going to be attached, some of those sugar molecules are going to be attached to each other and used to make glycogen and sort of stored away. So that's glycogen. This is cellulose. Cellulose is also a polymer of glucose. This is one glucose monosaccharide attached to another glucose monosaccharide. And if you attach them in this way over and over again, you make a polysaccharide called cellulose. This is not cellulite. I think that's the stuff that ends up on your legs if you get older or something like that. This is cellulose. Sometimes students get confused by that. The attachment here is a little bit different, so I want to point it out to you. You can see that it kind of has this zigzag attachment that the attachment goes up, then it goes diagonally down across this oxygen, and then it goes back up again. This is a way of saying, look, the alcohol group on this monosaccharide was locked in the up position, and the alcohol group on this monosaccharide, on this carbon, was locked in the down position when the attachment was made. And they're basically saying, look, the way that these things were pointing when the attachment was made is important. If you attach the glucoses when one of the alcohols is pointing up and one is pointing down, you make cellulose. If you attach them, and just to talk a little bit more about cellulose, cellulose is a polysaccharide that's found in grass, it's found in cotton. You can eat grass and you can eat cotton, you just won't get much energy from it. Your body does not know how to break this particular attachment to extract the glucose out of it. So we can't use that. We can't use cellulose for energy. We don't have the particular enzymes, the particular molecules that can break these attachments and get the glucose out. Just as a comparison, this is amylose again. And you can see this little U here. The U means that both of the alcohol groups were pointing down in this molecule and in this molecule when they were attached to each other, and compare that to this zigzag. So this tiny little change, the fact that one was pointing up and one was pointing down versus both of them pointing down, makes a huge difference in the type of molecule that you have. This stuff over here, you can eat it, you just can't get any energy from it. You can't eat it and sort of derive calories from it. This stuff down here is basically crackers, or one of the main molecules in crackers. And you can eat this all day long, and your body knows exactly how to break this attachment, and this one, and get the glucose molecules back and extract energy from it. So this is basically why mutarotation is important. Mutarotation is the flipping of this alcohol group either in the up position or in the down position. And you might look at that, and this was several slides back, you might look at that mutarotation slide and say, look, who cares whether the alcohol is pointing up or whether it's locked in the down position? It's such a little change. But that little change actually makes a huge difference when you start attaching these molecules to each other. If it's pointing one up and one down, you can make a molecule and you basically can't get any energy from it, or at least we can't. Or you can have them both pointing in the locked down direction, and we can't. Our bodies can use this stuff for energy. So that seemingly tiny little change makes a huge difference in the properties of the molecule, and it doesn't look like much, but it's an enormous difference. Cellulose, another feature of cellulose. I've been saying that polysaccharides are used for long-term energy storage. Sometimes they can be used for other things. One example of this is that cellulose can be used as a structural polysaccharide. What structural means in this case is it sort of gives a certain shape or rigidity to certain cells that make cellulose. So cellulose is made by plants for the most part, and what they're saying is that the cellulose molecules actually make the leaves of the plant molecules relatively stiff. And they sort of try to explain it this way. They say, look, if you could zoom in on a leaf, you could see this plant cell here that I'm sort of circling, and this yellow stuff on the outside of the cell is what's called the plant cell wall, and this stuff here is pretty rigid. And if you attach enough of these rigid cell walls to each other and make sort of this giant network that you can only see a little part of, then you will make the leaf relatively flat and stiff. And if it's flat and stiff like that, it's more efficient at capturing sunlight and getting energy from the sun, which is what plant leaves are pretty much designed to do. And then if you could zoom in on this cell wall that is making this plant leaf rigid, you'd see these little sort of tracks, which are called microfibrils. Again, you don't need to know this, but then if you could zoom in on one of the tracks, you would see many, many cables. And these cables, if you could zoom in on them, are basically chains of cellulose. So they're just these gigantic chains of cellulose bundled all together. They make these tracks. You line a bunch of tracks next to each other over and over again, and you make a plant cell wall, which makes the plant leaf relatively rigid. It gives the leaf a certain structure, which is why it's called a structural polysaccharide. If you've ever eaten lobster or crab, a large part of the lobster and crab shell is also made of polysaccharides that are obviously really tough and hard to break. So that's another example of a polysaccharide being used to provide structure to a living organism. So that is it for polysaccharides. What I would like you to know is I want you to know that there are different kinds. I want you to know that they can be either long chains or chains with branches. I want you to know that humans use glycogen to basically as long-term energy storage. And I want you to know why we can use some polysaccharides and cannot use other polysaccharides for energy. And it's basically that some polysaccharides are made with certain attachments that the molecules in our body cannot recognize and cannot break to release the monosaccharides. So that is it for polysaccharides, and that's it for this chapter and this unit. I believe next week is the last unit that we're going to cover in the textbook, so we're nearing the end.